Hello, everyone. On behalf of Carterra, we would like to welcome you to our webinar, Massively Parallel SPR-Based Fragment Screening on Ligand Arrays. My name is Cherie Salazar, and I'm the host and moderator for this event. If you have any questions during the webinar, please click on the Q&A button in the top ribbon next to the chat button and ask your question. We will get to the questions after the presentation. I'd like to introduce our speaker, Anthony Gianetti, who is the Associate Director of Applications at Carterra, working with our small molecule applications. Tony, I'll turn it over to you now.
Thank you, Cherie. So I'd like to talk today about the launch of our latest flagship product, the Carterra Ultra, which has enabled us to do fragment screening as one of our applications on a rather large scale that I'm excited to share with you today. So just as we get into it, a little bit of background on fragment screening. This is an approach to drug discovery that started to become more popular around the mid-aughts, and it was a counterpart to the idea of high-throughput screening, whereas instead of trying to screen upwards of 1 million molecules in activity-based assays, that we would instead try to look at the screening of just a few thousand very, very small molecules, usually molecular weight under 300 or fewer than about 17 non-hydrogen atoms, because the idea was that this would allow you to cover the full diversity of chemical space a bit more thoroughly than you would with about 1 million larger molecules. And so to schematize this process, the idea is that you try to identify these weak binders and then get structural information about them in the context of the proteins they're bound to, and try to find ways to then connect them together. So here we're showing a protein with a number of different fragment subspots highlighted in these electron density meshes from the X-ray crystallography experiment, into which models of the individual fragments are placed. In this case, multiple subsites being identified, and then from analyzing their geometry relative to each other, strategies of growing and merging or linking these fragments to grow them out to be very high efficiency binders proceeds. So this process works, and in 2011, the first fragment-derived drug went to market. This was Zelboraf, developed by Plexicon, who began with a structure of BRAF bound to 7-As and indol, and then were able to develop it through structure-based drug design into a potent inhibitor that had significant clinical efficacy in metastatic melanoma. This approach has been used numerous times, and as of 2024, there are now 7 marketed drugs that are derived from fragment approaches, and about 60 that are currently in phase 1 or beyond. So we'd like to, at Carterra, try to assist in the development of even more fragment-derived drugs. So the problem we face in trying to initiate a fragment-based drug discovery project is that you're trying to look for the ability to screen some thousands of compounds with a low molecular weight, with affinities ranging from, in the best case scenario, about a micromolar, but usually up in the hundreds of micromolar to even millimolar. So that means that biophysical methods will tend to be the preferred approach, and they're more popular than biochemical methods. And surveys of people participating in fragment drug discovery have shown that SPR has emerged as one of the most popular techniques, along with NMR and numerous other approaches, usually working together synergistically. So a quick bit of background about how SPR works. This is an experiment that begins with a gold chip that is being illuminated by plain polarized light at the total internal reflectance angle, and changes in refractive index near the surface cause that angle to deviate slightly. So we begin with the chip docked in a fluidic flow cell, microfluidic flow cell, where proteins of interest, or RNA or DNA or molecules of interest, are attached to these to a hydrogel schematized here by these long green strands. The flow cell, the microfluidic flow cell is then injected with a potential binding partner, and as that binding partner interacts with the matrix, you see a rise in response over time. And at the end, after a period of injecting what we call the analyte, the injection is switched over to just running buffer, and then we're able to watch the molecules dissociate in real time, shown here leaving. And so by this experiment and cycles of injections, we're able to see the real-time binding and dissociation of molecules from these surfaces. So the kind of information we can collect from an SPR experiment would be, as shown in the previous slide, the real-time ons and off rates, which once fit to an appropriate model by their ratio determines the binding constant. But in the fragment world, we're going to see a lot weaker binders, where binding proceeds to equilibrium almost immediately, and dissociation is almost immediate. But in those cases, by testing multiple concentrations and recording the binding level, we can fit a model to find out the KD from the midpoint of the transition here. And over many, many experiments by myself and many other users in the world, we find that in the affinities measured by SPR match very well the IC50s measured in biochemical assays, usually with R-squareds better than 0.9 for the cases where active proteins are available. So SPR is a great technique to support fragment-based drug discovery because it offers an enormous dynamic range in affinity, shown here in my hands as far as 20 millimolar in this case, and as potent as 50 picomolar in this case. So the instrument can really support and work over the full range of affinities of molecules seen during a drug discovery program. SPR is also extremely sensitive and reproducible. So here I'm showing an experiment where the lowest concentration is only occupying 6% of the total surface capacity, and yet its signal is quite visible well beyond the baseline noise. In addition, the real-time nature of the experiment allows us to also interrogate the quality of the interaction. And so not all molecules are single site, stoichiometric, reversible binders. We also encounter molecules that we call promiscuous binders or promiscuous inhibitors, which are molecules that aggregate and don't bind in a drug-like way, but often still act as inhibitors in enzyme assays. And so they end up as false positives in both screening and downstream hit to lead assays. And so SPR is able to readily identify that this, for example, this binder has way too high a signal, is appearing irreversible, isn't saturating, and doesn't look at all like the other three. So Carterra's approach to SPR revolves around the idea of creating large ligand arrays. And so we have a multi-channel flow system that docks to chips and creates individual flow channels onto which ligands can be arrayed. And then once those have been put down, the multi-channel head undocks and a single flow channel system docks, and then individual molecules, analytes are flowed over all those spots simultaneously. So on our previous instruments, the LSA and the LSA-XT, up to 4 different arrays could be made, allowing you to spot down up to 384 ligands. With the Ultra, we've taken that to 2 arrays of 96 for 192, up to 192 ligands total, or 191 with a reference. And the chip types here support a variety of capturing and chemical formats consistent with other SPR systems. So to show you in a bit more detail, the preparation of the ligand surface begins with this 96 needle manifold that sips into 96 or 384 well plates, draws the solutions up, and then creates on the chip these small flow channels, isolating each ligand individually, and they are passed back and forth over and over on the surface for a user-specified amount of time. So one of the great features of this is that your capture times can be essentially unbounded using only a small amount of ligand, generally about 200 microliters, but you can capture for a few minutes up to hours. And one of the great features is that it actually returns the ligand back to the source plate, meaning that preparing your surfaces is non-destructive. And so here I have an example of data where showing a amine coupling protocol where the chip is first activated in the single flow channel system, then the multi-channel head docks, and we pre-concentrate and couple proteins. I had several different concentrations here, but each of these is 12 different spots. each with different concentrations of protein. At the end of the coupling reaction, we then block with ethanolamine, which knocks some of the pre-concentrated material off and wash. And this allowed us to capture, anywhere from 5 to 20, sometimes more than 20,000 RUs on the surface. Once the printing is done, the single flow channel docks, and then very similar to the multi-channel side, analytes are drawn through, and they are also passed through the single flow cell in this back and forth pulsing manner. And this will repeat from a different angle. And so one of the nice features of this instrument is that the amount of analyte solution that you use is the same regardless of whether you do a 30-second injection or an hour injection. So the amount of sample is the same regardless of duration of your association phase. So this is what the multi-channel print array looks like for the Ultra. To try and get the best possible data for small molecule applications, we've gone ahead and arrayed the reference spots right next to the target spots. So after making the chip, you'll have 96 reference spots, 96 target spots. We can also use some of the reference spots to put down additional targets, and we'll show an application of that, allowing you to do, again, as I said, up to 191 with concomitant decreases in data quality, but they tend to be pretty small changes. And then once the analyte single flow channel docks, the analyte is flowed in this direction over the array. So I'm still getting used to the amount of data this instrument generates. And so we look at the data in these grids, 12 by 8. And so a single dose response of an analyte ends up generating nearly, in this case, 768 curves for a 200 Dalton compound binding two surfaces with Rmaxes ranging from about 5 to 10. And so it's a lot of data to ingest. But if we go and take a closer look at one of these, we'll see that in this lowest concentration injection, we actually get very clear binding that fits nicely to a one-to-one model under 1 RU of binding. So the Ultra really has incredible sensitivity and low noise for detecting small molecules. To wit, I'm showing here the fit to 96 different surfaces with different ligand densities, the same 200 Dalton molecule, and I'm showing the residuals, which for me is where the proof in the pudding comes from, that this is, again, 768 double referenced injections overlaid, and we can see that there's no shape to the residuals, that all of the scatter happens, almost all the scatter is between plus or minus a half RU, with the rest of it coming in between plus or minus 1 RU. And so this tells me that all of the work that the engineering and R&D teams have put in to manage thermals, to manage noise, to make the fluidics and the transition super smooth, have converged to make an instrument that produces very low noise, very reproducible data. So to try and do some science with this and to move into fragment screening, we partnered with Carna Biosciences. They're a company that produces, has about 20 years experience in producing kinases for uses in different applications. Actually used some of their very first biotinylated kinases back around 2008, 2009. And they were great then, and they're still phenomenal. So Carna offers a variety of services, including assays and cell-based screening and selectivity profiling. But they also commercially offer a wide variety of human kinases spanning all the kinase classes, and they make them available in different tagged formats, including GST and HIS. But what I'm going to talk about are their biotinylated kinases. So they offer about 600 different kinases, 2 of them biotinylated, and they offer about numerous assays for characterizing those kinases. So Carna expresses the kinases in an insect cell system, and Affinity tag purify them, and they provide testing information on their website for activity from various biochemical formats, mostly nanobret, and also increasingly SPR, including data now from Carterra. The kinases are expressed with a biotin acceptor peptide as part of the protein sequence into which a single biotin is added. And for every batch, they confirm that the in vivo biotinylation efficiency is north of 95% to 100%. So one of the nice things about the Carna portfolio is that they also offer kinases in multiple forms, specifically the inactive conformations, but they also take pains to activate proteins where they can express with upstream activators or do in vitro activation with ATP for enzymes that auto-activate. And they even have a number of stable complexes, such as the CDKs, a number of CDKs with cyclin, where we've now been able to show on chip that even though only one of the two proteins is biotinylated, the complex holds together very nicely on surface and is not seen to dissociate. So having this kind of reagents available is fairly important in kinase drug discovery, where of course one of the challenges is trying to provide selectivity across the greater than 400 kinases in the human genome. And one thing that has been seen again and again is that it's often the case that you can make a more selective molecule to the unactivated form of the kinase than to the activated. Once activated, kinases tend to look a bit more like each other than in their unactivated state. And so this is one of a couple of programs I participated in. We're making a compound that bound very, very tightly to the unactivated form of the kinase, and much better than to the activated form, produced less toxicity and fewer off-target effects than compounds that bound equally well to both of them. And I'm schematizing BTK in particular here, showing the variety of conformational states that it can undergo with these large swings of the glycine-rich loop, the C helix, and the two halves of the activation loop, which really undergo dramatic changes and affect greatly the shape and properties of the ATP binding cleft, which is where most drugs are directed towards. So having this diversity of states of kinase as well as a broad portfolio of them really helps with trying to initiate and understand selectivity in drug discovery. So to schematize how we use the kinases, this is an example of doing a capture of 96 kinases to one array group on the chip, followed by an additional set to a second array group. Carterra provides pre-made streptavidin chips that are able to capture to very high densities. So that the highest ones in here are in excess of 20,000 RUs. These chips are pre-coated with streptavidin, which binds biotin at a high affinity. And you do need references. So in this case, we got 96 targets down, additional 29 on that, using some of the reference spots, but still giving ourselves a nice number of references. So a preliminary experiment to evaluate how well our technology was working with these kinase arrays, we started with a smaller portfolio of about almost 90 kinases and the Tokris library from Biotechne of known kinase inhibitors. And these were run, this was the entire experiment shown here, where each segment is a dose response on these almost 90 kinases for all 200 compounds, with a control compound intermittently run to verify that there was still plenty of surface activity available and binding sites, that we weren't binding up too much protein and losing sites. And so there's an enormous amount of data in this run, and we can look at a number of the features that came out of it. So for example, looking at kinase selectivity again, we see here on the left a compound that binds many kinases, though with quite different affinities. We see some very potent ones, we see some very, very weak ones, but that stands in contrast to this compound, which really only bound a small handful of kinases, and only some of those fairly well. So if you're familiar with these kinome trees that are produced by a number of different third parties, where you plot the percent inhibition of a biochemical assay against the location of each of the 400 or so kinases in this dendrogram representing the the phylogenetic relationships between the kinases, we see that if we repeat this using the KD values that come from these experiments, we get essentially the identical array and distribution of sizes of spots. So this suggests that our platform could be used to do broad kinome profiling, similar to the kinds of work that's done. In this case, Karna provided the assays to produce this tree. So we then moved on to performing a fragment screen using the Maybridge 1000, a commercially available library curated for solubility, purity, and diversity. And we made a chip with 125 of the biotinylated kinases from Karna, and then proceeded to do a screen looking to select positive binders, which will move on to affinity determination and ligand efficiency determination. What I'm showing in this video to the side is essentially a representation of how fast the instrument is. So our cycle times for analyte injections are right around 3.5 minutes in a fragment screening mode, but each injection, of course, gives you information in this experiment of 125 different proteins. And so this is basically the time average real-time data collection rate of fragment protein interactions when done with our approach using these large arrays. So here's the outcome of 3 days of screening. And I should say this was all done by a single individual running the instrument and doing all of the buffer preparations and plate preparations, et cetera. And so what we're looking at is 125,000 interactions, where in this panel, we're looking at every color is a different kinase. So you're looking at a complete 1,000 member screen for 125 proteins. And up here, we're looking at the fragments in red and the controls AMP&P, which was AMP-PNP, shown in blue. And you'll see some variation in the degree of AMP-PNP binding. There's a number of kinases, especially some in their inactive forms, that won't bind ATP until they're activated by upstream kinases. And we see that reflected in which kinases are not showing a lot of AMP-PNP binding. So that's expected behavior. What I've highlighted is one hit, one particular hit of the many that are available here. Just highlighting that in our hit selection, we are getting the kinds of fragments we would expect. In this case, a difluorophenyl aminothiazole binding to check 1 in the expected manner. Our software also supports the export to third-party tools. So I'm showing here the cheminformatically aware program Data Warrior, which is freely available, and that it did a nice job ingesting this rather large data set and connecting it to the chemical structures. Within our software, though, you can certainly interrogate the data. And so here's an example of WEE1 kinase from the larger set, showing the AMP-PNP replicates. And you can very clearly see just by eye that there's a number of hits popping up above the noise. And so we can go and interrogate them, find out more about their identities, and move them on for further downstream characterization. We are in the process of implementing additional tools to help with automated data analysis. In the case of fragment screens, there is sometimes a little bit of drift, daily drift in the controls, drift in the baseline, and also different binding levels from different days or different kinases to the controls that are being used. So we're implementing methods that will allow the scaling, the data correction, the scaling of the data to be put on a common scale so that selected groups of hits from across the array can be compared to each other to try and get upfront information about selectivity and specificity for whatever group of proteins you choose. We're also implementing statistically robust approaches to hit selection, in this case using norm exponential deconvolution, which separates the Gaussian distribution of non-hits from the tailing exponential of positive hits, allowing very good identification of the difference between the noise level and the binding level of hits. So we've also this will be talked about at a later date, but we've also done a large fragment screen with John Quinn from Genentech, looking at 92 proteins on about 3,500 compounds. And so a bit longer screen, but in 11 days, over 320,000 interactions were measured. And so more will be talked about this, but this is the kind of fragment screening we're starting to do with the Carterra Ultra instrument. So this is the Carterra Ultra. It's got a number of different enhancements, as I said earlier, thermal, optics, and a number of actions to help it run faster and get shorter cycle times, and really high quality reproducible data, working in not just small molecules, but also in proteins. And so this is some data collected by one of our in-house scientists the other day, showing 53 overlaid replicates in an epitope binding application. But the important thing to notice is that all of that's done under 5-hour use of binding with great reproducibility. And so with that, I'd like to thank a number of people, especially Rebecca Rich here at Carterra, who has carried out all of the fragment screens so far, Karna Biosciences for providing a lot of kinases and guidance on their use, Biotechne for the Tokris library, and Maybridge for the fragment library that we've been screening. And with that, I'll open it up to any questions. And so I see some coming in. Keith Pitts asks if we can address the challenges of aligning conditions for simultesting of multiple targets. So good question. Yes. One thing that I've noticed over time is that a lot of assays seem to be converging on a lot of fairly similar conditions. And so, yes, there is the challenge of trying to work with this many proteins under a single buffer condition. But I think a lot of what we found, especially with the kinases, is that a fairly simple, straightforward buffer of 50 millimolar heaps, 100 millimolar sodium, 150 millimolar sodium chloride, 5 millimolar mag chloride, some detergent seems to actually work pretty well across a lot of them, and we see pretty good activity, sustainment of activity across surfaces. That will be an ongoing challenge that people will have to continue to address to use these large arrays. Keith also asks, are the samples pulsed back and forth examples for the fragment kinetics we showed, or were those injections more classic? Don't know exactly what you mean by classic. The instrument always does these back and forth pulses. So even if it's a 30-second injection, it'll pulse back and forth. There's only about, I think, 20 microliters, 70 microliters, something like that in the actual flow cell. And the sample volume pulled up by the needle is about 180 microliters to fill the fluidic paths. And so whether it's an hour association or just 30 seconds, which is our shortest injection time, those pulses will always be used. Let's see. Do we have any additional questions? Okay. Well, if you think of additional questions in the future, please feel free to send them to questions@carterrabio.com, and they'll be answered either by myself or we'll connect you with one of your field application scientists in your territory. Sheree, I'll turn it back over to you.
Thank you, Tony. That was a great presentation. As Tony mentioned, if you do have additional questions after today, please write to us at questions@carterra-bio.com. Thank you all so much for attending this webinar, and have a great rest of your day. Bye-bye.